And for those who enjoy this channel and would love to support us financially, please feel free to hit that donate link. We'd greatly appreciate it. God bless.
Hi guys, we're back. Thank you for the intro, Raj.
I just wanted to show it off.
Yeah, and it was great. I loved it. We all loved it. We laughed, we cried, we were moved. So we're gonna today's debate is: does genetic analysis support evolution? Did I get that right, Catz? Did I get that right, Catz?
I think so.
Yay!
I think more or less. I think more so. Does genetics confirm, or is it more consistent with evolution or creation? Because I'll have a position to defend as well. I don't want to make poor Catz, the only one who's defending a position here. So
Cool.
So before we start, let me just thank Mistake Not for £2 oi oi super chats too. Did I say oi oi right? No, I'm kidding.
Oi oi.
Tequila, the cool. Oh, that's a cute sticker. Thank you. And C4, be safe, everyone. Yes, for $5. Thank you very much. Be safe, everyone. Yeah, please be safe out there, guys. I love y'all, and you mean a lot to me. So, let's go. We're gonna start with each one's gonna make 5 to 10 minute opening statement. Who wants to go first, guys?
Who goes first? Because I know he's been a bit rushed, so I'll let him decide who goes first.
it's totally up to you. I mean, I've gone first. I've gone second. What makes more sense for you, Catz? I mean, I can start if that's easier. It's up to you.
How longer?
I don't mind. Toss the coin.
Okay. If you want to start, you could start, and then I can. Yeah, it was a little bit rushed there, so I'll gather a few things and get ready.
That sounds fair. Rajiko, before you start the timer, can you get my first slide up, just the cover title slide thing?
Sure.
Is that okay?
See? Yeah, I got your screen right now. I need to activate this one. Oh, there we go. And your first slide is this one.
Right. Okay, I can't quite see it up on the screen yet. So, nerves now. Give us a... No, not that one. Not that one. The one before. That was my big entry. The title one.
The title
one. Okay.
Yeah.
The title one.
Hey, what do we pay you for?
You had one
I'm getting paid.
One job.
That's why we pay $100.
Right.
I always say that to my tech guy. I said, you had one job, and you couldn't even do that.
Right?
I'll remind you guys, I have a really big sword.
He's
So
listening.
They'll run you through.
it's a 10-minute counter. Is that the plan?
10, yeah.
Up to 10 minutes. Up to 10 minutes, right? Yeah. So we could add anything they don't use on to like the end of the questions and answers or something like that. Whatever they want to do. Oh, Simon Dan's here. Hi, Simon Dan.
Hey, Dan.
He came for Catz.
Tell me when it's time to go, and I will go.
Okay.
I will push the button now.
Reg.
Okay, so what I want to do is begin with the science of genetics and how it makes predictions that will be completely for evolution. Now, before genetics appeared, we had fossil records, geology, radiometric dating techniques, and that had all set an idea in our head, a conclusion in our head, that humans were not unique creations, but were descended from a different type of organism. To the conclusion that we were very, very closely related to the chimp. Okay, but then along comes genetics, and it instantly gives us a problem. Next slide, please, Raj.
Will do.
We got it, yeah. Because the problem was, it turned out that chimps have 24 pairs of chromosomes, and we as humans, we only have 23. So this is where science has to make a prediction. We have to make a prediction for we have to use our scientific knowledge to make a prediction about what's going to happen next. And the two things that are going to happen next is genetics is either going to show us that a chromosome in a chimp on some sort of fission or fusion has occurred in a human. Now, if you go to the next slide, Raj. To understand the significance of this, we've got to understand what a human chromosome looks like. And we can see that the centromere, it's not in the middle. That's like an area of highly specialized sort of chromatin. It plays an important role in mitosis and other things. We refer to the bits above and below it as centromeres or arms. But essentially, looking at that slide, we can see that if we were to take 2 of those chromosomes that you can see on the left, and we were to stack them one on top of the other, we will get the image that we see on the right-hand side. We will get one long chromosome with 2 regions of centromere, would have telomeres at the end, and would have a telomere section in the middle. A little childish drawing I've done on PowerPoint. So we look for something that looks a little bit like that. Next slide, please, Reg. So science, if evolution was true, would predict we'd find something like that. And this is exactly what we see. Are we on that next slide, Reg, now, actually? Because there's a bit of a lag.
Oh, yeah, we are. Yeah.
Yeah, so this is exactly what we see. And what we can see, though, is the human chromosome number 2, chromosomes 12 and 13, and instantly you'll see there's a perfect match between the banding patterns. And when we used our predictions to find out where the lost or the remnants of that centromere, the second centromere should be, we can find it. We find it in that Q21.3 to Q22.1 region. I've highlighted that on the screen. So we've used genetics to make a prediction, and that prediction has come true. Now, that's not something creation can do because chromosomes aren't even mentioned in the Bible. Predictions end, they keep going. And the next one is sort of an absolute belter. Next slide, please, Reg.
Yeah, there it is.
So we want to really hammer this home and make sure that this really is the fusion of a chromosome or 2 chromosomes into one. And I want to talk to you very briefly about a telomere and what a telomere is. You see, when cells divide, chromosomes are replicated, we lose bits off the end of the chromosomes. Now, to stop the genetic material, the information being lost, the ends of the chromosomes are long repeats. And I've put the repeats on the top, TTA, GGG, or the mirror of those, which is triple C, T, double A, because C's go opposite G's and T's go opposite A's. Long strands, and they have 2 jobs. One is to stop chromosomes from fusing. And the second one, like I said, is to protect loss of information, because when a chromosome is replicated, bits of the telomeres are lost. Now, important to know that when those bits of telomeres are lost, an enzyme called telomerase will come around and it will add more of those repeats. All right. Now, this is going to become important later on. So, if 2 chromosomes have fused at the telomeres, then we should be able to make a prediction, and that is in the center of chromosome 2, fusion's taken place. We're going to find lots and lots and lots of these TTA, GGG bases. If you can go on to the next slide, please. Now, this is the actual fusion zone itself, and highlighted in yellow is the suspected point where the specific fusion took place. And it's exactly as we would expect. However, oh, you seem to have gone backwards there, Raj, not forwards. No, can you go on to the next one? I've been, yeah, I'll put the wrong slide on. The next one after that, the one that shows all the letters. So what we've got here is the actual fusion zone highlighted in yellow, and you can see around it all of, you might get it on my screen as well, we can see lots and lots and lots of TTA, GGG, and lots of the mirrors as well, which is what we'd expect with a telomere. But even more, there is another prediction here that evolution would make. You see, I said before that telomeres are there to prevent chromosomes from fusing. So how could a telomere fusion take place if that telomere was fully functional? Well, it turns out that telomeres can if telomerase isn't really working, there are studies that show telomeres can time, and when they degrade over time, we get what we see here. We don't get perfect repeats of TTA, GGG, right? What we get is something that looks highly like it, but with little differences sort of in between. Now, I want to point out here that there's a 1 in 4,000 chance of a sequence bases of a TTA, GGG. There's a 1 in 4,000 chance of that happening randomly. So we can see that that is certainly not a random bit of DNA. Marks of a telomere that's degraded over time. Now, we'd also say that because we understand that telomerase will kind of freshen up the telomere by adding more of these sequences, it will only do that on the ends of the chromosome, not in the middle of the chromosome. So this has been left degrade to the point that it's degraded at. Again, it's another perfect prediction of evolution, and it's not something that creation would ever talk about. We got time to make more predictions about evolution? Well, yes, we have. Can we go on to the next slide? Because evolution, it's the one that says 8. Yeah, it says 8 on the PowerPoint. It's one that says it's not just humans at the top. So we would predict that this wouldn't be a unique event. We would find it in other animals. And when we look at pigs, we can find 2 different types of fusions in pigs when we compare it with a close relative, the babaroos. So we can find that chromosomes 3 and 6 have also fused, one in a telomere-telomere fusion, the other one in a telomere-centromere fusion. So we see that, and we should, if this is correct, see this in lots of different places. So if you go on to the next slide for us, please. We do. All you've got to do is spend a little bit of time looking on the internet, looking for telomere-telomere fusions, etc., to find lots and lots and lots and lots of papers. And they're not all historical. I know creationists might say these things happen over a long period of time, so therefore you're just giving me historical stuff and you don't really know what's going on. Well, one of those there, the middle one on the left, the spontaneous telomere-to-telomere fusions occur in yeast cells. If you go on to the next slide, Raj. Which has been observed in the lab, fusion in plasmids in a yeast cell. And if you go on to the next one as well, please, Raj. Something else that's been seen in a lab is when mice have been treated so the telomerase is no longer functioning. It only took 6 generations before we were actually getting telomere to telomere fusions in the lab. That's been seen. Now, I was hoping to be able to get through what my response to common creationist arguments will be about this, but looking at the timer, I'm not going to have enough time, so I'm going to use, I'm sure that will come about in the discussion that we have. But ultimately, chromosome 2 has made a series of predictions that have been proved to be correct, predictions that creationism cannot make because there's no mention of chromosomes or DNA or genetics in the Bible. Sort of personal incredulity, I really have no idea how this can be refuted. I know we're down to the last minute. I'm nearly done. I know that Standing for Truth is going to talk about Tompkins, and he might want to talk about the DDX 11L2 gene, etc. And I'm more than happy to discuss that. But for now, I think I've laid a strong case for humans being very closely related to chimps. And I'll stand down.
Thank you. Wow. That's great. That was great. Thank you very much. Standing for Truth? When you're ready.
Oh, I promise I didn't run away, just on mute. Okay. No problem. Thanks for that opening there, Conspiracy Catz. I appreciate the visuals as well. Very informative, and yeah, good job. So what I'll do is I'll set my timer for 10 minutes as well, and make sure that I don't go over. So I'll start it now.
Do you want like oh, sorry.
Oh, no, that's okay.
I was just going to ask if you wanted like a little boop, like, 30 seconds before the end or something.
Maybe give me a 1 minute warning. Great. Because once I get on a roll, I may lose track of time.
Great.
Okay.
Thank you.
Perfect.
Thank you.
Perfect.
Thank you. Thank you very much.
Okay, thanks for giving me the opportunity to discuss creation and evolution today with Catz, with a focus on genetics, of course. In Katz's previous debate with Kent Hovind, he utilized genetics as a way to determine whether or not universal common ancestry is true. It's genetics, it's in our DNA, and it looks like Catz understands this, that, the question regarding ancestry is answered. Sperm and egg, they don't pass on a fossil or a bone. They don't pass on geography or rock. They pass on genetics and traits. And so if we want to find out the history of humanity and the origin of species, this is where we must look. And this is why I'm more than happy to discuss creation and evolution in light of genetics with Catz. The entire junk DNA paradigm, for example, has been overturned. Orphan genes show limited ancestry. How does Catz here explain this incredible class of taxonomically restricted and essential genes? The chromosome 2 fusion, as he spent his entire opening on, has been overturned. That alleged site where the fusion supposedly took place actually represents a highly organized functional gene. The area with the telomeres is far too generate. The second deactivated centromere should be found, but there's difficulty finding it. Maybe Catz can point that out to me in the discussion. But nonetheless, it's agnostic to the debate as a whole, because even if there was a chromosome 2 fusion, it is indirect evidence, as it would have had to occur in the human line. So it's not going to solve the answer to today's question. So endogenous retroviruses as well, which I know he likes to talk to, ALUs and other classes of retrotransposons, and even pseudogenes are consistent with the created heterozygosity model. I'll touch briefly on that here, but I'll be more than happy to explain in more detail in the discussion. The question we should be asking is, would God have created Adam with 2 identical versions of DNA according to our model, and then Eve the same way? If this is the way God chose to do things, Adam and Eve would have created clones. God said, be fruitful and multiply, not be fruitful and clone yourselves. What makes the most sense, both scientifically and theologically, is that God created DNA differences from the start. This is the idea of created heterozygosity. Adam and Eve would have had within their DNA the genetic potential to produce every shade of skin that exists on this planet or known to man. A man and a woman today could have thousands, even millions of children, and no 2 children would be 100% identical. This is because they are heterozygous. There exists built-in diversity in millions of sites within the genome in the average person. In the limited time that I have here, the best way I can explain it is, if God created Adam and Eve to be heterozygous, which in fact would be optimal design, you could have as much human diversity and more than we have today in their first family. Time is not required for the diversity today, because diversity can be designed and built into Adam and Eve. Catz also talked about predictions and how evolution can make predictions. Evolution has so many failed predictions. For example, the chromosome 2 fusion, and we're making predictions based on this hypothesis. We're making predictions on mutation rates. We're making predictions on DNA function. We're making predictions on speciation rates as well, and some have already come true. I'd be more than happy to elaborate on that in the discussion. He talked about fossils and dating, of course, as I indicated. Rocks and fossils, they're not passed on sperm and egg. Traits and genes are. So that's why we're here to talk about genetics. A few questions I have for Catz regarding this topic would be, how does Catz explain the incredible dissimilarity between chimp and human Y chromosomes? If the chromosome 2 fusion is agnostic to this debate, we need to look at some differentiating evidence. And if humans and chimps, as he indicated earlier, if chimps are our closest ancestor, why is the Y chromosome, which is immune to recombination essentially, so dissimilar, less than 70%? That's a question I have for Catz. How does Catz also explain just some of the HapMap data that tells us the human genome is young and shared blocks of DNA are large? This means the genome is young. Why? Because there has not been enough time to scramble them to randomness. How does Catz explain the low genetic diversity in human beings? Evolutionists invented the out of Africa population bottleneck that reduced the population of that time to just 10,000. Some even say 2,000, in order to explain low human genetic diversity. Is this not simply a post hoc and ad hoc rescue device? The low genetic diversity, the mitochondrial DNA, and the Y chromosome DNA variation all demonstrate irrefutably that we came from not just a small population, but a population of just 2, Adam and Eve. How does Catz address this evidence? Even if there was a chromosome 2 in the human line, it could have happened at the population bottleneck after Noah's flood, or even before, for it all to be passed on. Like I said, we need to look at the differentiating evidence. The evidence for the existence of Adam and Eve and biblical creation is you and me and the genetics of the people in the world today. The genetic structure in humans today speaks to us of a literal Adam and Eve. We have a small DNA compartment called the mitochondrial DNA that we only inherit from our mothers. Everybody on this planet got their mitochondrial DNA from a single woman, who is exactly what we would expect from the biblical Eve. She is quite literally the mother of us all. In a nutshell, we have nearly identical mitochondrial DNA, and the very few mutations that differ from Eve's sequence are exactly what would be expected if Eve arose just thousands of years ago. The same thing applies for the male side. In our DNA, we have a piece of DNA that we only get from our fathers, if we are male, of course, and this is called our Y chromosome. The Y chromosome is passed on unbroken from father to son. Every once in a while, a mistake happens. And every time this happens, a new branch in the family tree is formed. If we look at all the branches in the world, they go back to a single person, not a chimpanzee, sorry, Catz, but to a man who lived just 1,000 years ago. A few thousand years ago. This is Y chromosome Adam. The evolutionists respond by saying, oh, these aren't the Adam and Eve from the Bible. These people go back hundreds of thousands of years ago and they're part of a population. They are not using observed mutation rates and are using evolutionary phylogenetic calculations and assumptions. We are using the empirical method. I'm more than happy to discuss this with Catz. As a matter of fact, the data is so strong and conclusive for the Y chromosome that a new research paper confirms that if humans have been around for as long as deep time evolution would suggest, then they would have accumulated 8 to 59 times the amount of mutations that we currently observe in Y chromosome DNA sequence. The empirical data suggests that we only observe approximately 4,500 years of mutation accumulation in the paternal ancestry that is contained in the record of this human Y chromosome. How does Catz deal with this? What type of selection, for example, can Catz present us with that will select away so many deleterious mutations that are pouring into our genetics and degenerating our information systems? If he wants to use indirect evidence, such as a possible chromosome 2 fusion, be it he can't address the problem of genetic decay, then it's nothing but indirect evidence that would need this so-called problem of genetic degeneration explained. Most mutations, the fact is, they are unselectable and invisible to selection, and artificially contrived rescue mechanisms such as synergistic epistasis and mutation count mechanism have been falsified. Can Catz answer any of these? We will see. I got 2 minutes here, so I will just go over a couple things, a few examples that evolutionists will often use in not only DNA, but also in anatomy and physiology is that groups within groups patterns that they point to as evidence for descent with modification. Nested hierarchies in DNA, for example, anatomy and physiology, are also predicted by creationists based on the design model. The classification of life can be compared to the classification of modes of transportation on how different companies manufacture cars based on similarities, forming hierarchies. Naturally, for example, we as humans have designed things with groups within groups patterns. If you were to look at the types of vehicles or modes of transportation humans have created, they naturally form groups within groups patterns. A sedan has a tremendous amount of similarities with other sedans. Now, a semi-tractor trailer is obviously much more different than a sedan or a car, but these two modes of transportation are similar enough to each other that it sets them apart from airplanes. This is the groups within groups patterns that emerges when we look at all the modes of vehicles of transportation that humans have designed. This means we too would expect humans and chimps have a lot more in common and a lot more in genetics, in anatomy and physiology than, say, the old world monkeys or fish or bacteria. This is just common sense. Now, we have lots to discuss, but one line of evidence Catz has used as a way to demonstrate descent with modification in his previous debates and on his channel is endogenous retroviruses. And I believe Catz deserves a solid answer to this. I haven't seen a solid answer given to him yet, so I'm more than happy to discuss ERVs and demonstrate why ERVs and other classes of retrotransposons is simply just one more line of evidence consistent with the created heterozygosity hypothesis that I've briefly touched on. Thanks so much, guys.
With 10 seconds to spare.
Just
wow.
Time.
All right, well done, guys.
Yeah, just from the opening statements, this is amazing. This is going to be great. So we're going to head into the 15 minutes or so of just a discussion between you two. Do you want to do like that? Is that good for you guys?
Yeah, I think we can get on.
All right.
I've just talked a lot, so if Catz wants to start it
Open
off
up.
and give his thoughts, I'm happy with that.
All right. Well, what I want to do is make sure before this debate is over that I haven't ignored any of the questions you've got. All right. So I know you've just done a big, long then, but if you just rattle off 6 or 7 of those things you want me to get back to, and I'll make sure I will, but I wouldn't mind starting off with chromosome 2 again, if that's okay. But if you rattle off 6, 7, we'll go over those.
Absolutely, yeah. If chromosome 2 fusion is where you'd like to start, then I'm happy with that. No, I guess my question would be regarding human chromosome 2, since it is indirect evidence for human evolution anyways, as I'm sure you'd agree, it would have had to occur in the human line when the chimp and human line separated from a common ancestor roughly 6 million years ago. So how would you say that is, direct evidence for ape to man evolution? Take your time.
Yes, all right. Well, I think when you're looking at evidence for evolution, especially genetic evidence for evolution, if we just look at one little box and then another little box and another little box, and we talk about this particular ERV or this particular chromosome, etc., then we're missing the big picture. The big picture is evolution would make a prediction that we would find a fused chromosome, which we have. Evolution will make predictions about ERVs. Evolution will make predictions about so many different things, and it's the weight of that entire body of evidence, if that makes sense. So I understand where you're coming from. Your stance is what essentially God created us with our chromosomes, and ours just happened to fuse, from being unique humans. Is that your standing point?
I would say, just based on my model of created diversity, or as I put it, the created heterozygosity hypothesis, God would have created Adam and Eve with pre-existing functional DNA differences. That's why we would predict that the vast majority of our DNA is functional, versus evolutionists that would say the majority of the DNA is based on genetic scars, genetic fossils, the infestation of parasites and viruses, of course. So that's, a head-to-head prediction that we can discuss and see what the data tells us, because all of that evidence, chromosome 2 fusion, endogenous retroviruses, pseudogenes, for example, or nested hierarchical patterns in anatomy, physiology, and genetics, if I can demonstrate that the vast majority of our genetics is not the result of deep time evolution, but is actually functional, then that turns those nested hierarchical patterns into evidence for design and not descent with modification. Go ahead, Catz.
Okay, right. Well, in that case, then I will address what you said about chromosome 2 having I think you said it had been overturned, didn't you? Do you mind if I just show a few slides, then you can respond to what you want? Is that okay?
Sure. Yeah, I'll look at your slide from my cell phone because I can't see it from the actual screen. Just one sec. Yep, go ahead.
Okay, so Raj, it's the slide that's like the 12th one down. I'd like to start at. That's okay. While you're doing that, I know there's a lot of people in the chat that follow the flat Earth scene, and a lot of you will be familiar with Ruhif. Now, I had no idea until I was researching for these debates that Ruhif is brilliant in this field. He has his own blog. He's got amazing videos on it, and some of the stuff, if Ruhif watches this back, I've pinched some of the stuff from him, and I just wanted to credit him for it because I forgot to label all the slides. So that's not there. Just 2 before. Where are you? Which one are you on there, Brad? Yeah, 12. Sorry. Yeah. A lot of the sort of overturning evidence, if you like, that has come out of the institute. Well, it's come up from the creation side against the fusion of chromosome 2. Comes from the Institute of Creation Research. Now you'll have a look at the bottom where I've highlighted it for you, and it makes it very clear there that they're not an independent science body at all. What they're there is to try and put God's word into what they can say. So they're not. I would say that they are the least sort of neutral body that I could possibly find to write a scientific paper. And it's interesting that Tompkins, who writes for them, unless I'm wrong, I don't think he's ever had a paper written, published by a mainstream scientific journal. However, if we skip on to the next slide, please, Raj.
Well, he has. I just want to correct you there. He has had secular publications. Before he started working at the ICR, I believe he ran a genomics research center. I'd have to double check exactly what he ran, what he did. But yeah, he's got quite a few secular publications.
Brilliant. Oh no, cool. Thanks. Right. So this is sort of the argument that he's given. And you know what? I'll shorten this down, and we'll just jump to slide 16. I'll just get straight into the nitty-gritty of it. We go on 16. So this, I have credited Ruhi for this. This comes straight from his blog, actually. This is a breakdown of that fusion zone. Now, on that fusion zone, it's claimed there's a gene that runs right across it, the double DX 11L2 gene. Now, in truth, the gene doesn't run across the fusion zone. Some people would suggest that if it ran across the fusion zone, then it couldn't be a fusion zone because they were claiming that two telomeres couldn't come together to create a gene. But actually, if anyone Googles the term fusion genes, it's quite a common thing for fusion to result in a gene. But either way, the gene in question, I'm just going to call it the DD gene because I'm not reading out the whole code every time. This gene is actually set to one side. Sorry, yeah, it's a mouthful. It's actually set just to one side of the fusion zone. The fusion zone is a promoter region. Now, a promoter region is a region where basically transcription of a gene starts. So you get transcription factors binding to it. And then they will read across the gene, and they'll read across it from right to left, as you see it here, and they will transcribe the gene. Now, this gene actually has 3 different transcription points, and the strength of those transcription points, how well they are used, is represented by the green on here. So you see the little green peaks, and the one that's underneath the red is the transcription point, which is on the opposite side of the fusion point to the gene that we're talking about. And the creationist argument from a lot of people is that if this promoter region, if this region of, this promoter region, if that can cause transcription across the gene, that's not a result of fusion. Now, actually, we need to look at, is that promoter really designed there for this gene? Is it? Well, the height of those green sort of hills are an indicator, if you like, of how efficient, how good a job that section is at binding to transcription factors. Is it good or is it not? And the higher it is, the better it is. Now, that looks quite good until you go on to the next slide. Can you go on to the next slide? Now, on this next slide, we're going to see lots of green spikes and lots of green things. And these green things are all the relative strengths of transcription factor binding sites from chromosome 2. If we compare that to the one which is on the opposite side of this fusion zone, I've had to put an arrow because I don't even think the red dot to give us the height even shows on this screen. There's a tiny, tiny red dot, and I've tried to blow it up next to it.
It's really small. I can zoom in. Let's see.
It is hundreds and hundreds of times weaker than all the other transcription factor binding sites on that chromosome that we're looking at there. It's not meant to be binding site for that gene. They are not one and the same. They are just coincidentally slammed together by that fusion. Otherwise, what God has done is he's made this fusion happen and then left as evidence to suggest that actually, it was, well, I don't know why he would do it. But anyway, nip onto the next slide. Is that okay?
Sure.
Did you want me to do like just a really big response after you're done, or did you want me to like address certain points that you've said already? Like, I'm totally fine either way.
Well, I'm about a minute from finishing, and then you do a big response. If that's okay, you do a big response. I'll keep my gob short, and is that this is probably the longest bit I'll be going for. Is that okay?
Going, yeah. As long as I get equal time to respond. I'm just writing down.
Can I just say how wonderful it is to see people like this? I'm sorry, but this is a great debate. You're acting so nicely to each other, very, polite and well mannered, and I love that. Thank you.
I haven't had to.
No problem. I'm enjoying. Hey, there's no reason why 2 people who disagree can't have a friendly discussion over it. That's for certain.
Absolutely.
Yeah, I haven't had to step in once. It's been great. Right?
Yeah. Well, if we then ask ourselves, there's that promoter region there, and if somebody's claiming that promoter region somehow is linked to that gene and it should be part of that gene, well, should it really? Well, subtelomeric DNA is DNA which is just offset from the telomere, and we can see here, and again, I think this is from Ruhif's blog. On the next, you can just cycle over the next 2 or 3 slides if you want to. Raj. What we find here are for other genes, promoter regions, which are also found in that kind of like subtelomeric region. So there's no reason that promoter gene couldn't have been part of the original telomere. And the fact that it's so weakly performing compared to all the others for this particular gene really sort of kind of suggests that this fusion really has occurred. And that was just some remnant in the subtelomeric DNA. So, whether there's anything else I was going to say about that, I can't think, but I'll pass it over back to Standing for Truth. But I think that's confirmation of the fusion.
Of course. Yeah. Thanks for, and once again, I always appreciate the visuals. So I would just say if I was a forensic investigator, for example, and I walked in and saw, 100 pieces of evidence that looked like somebody did it, all I need to do is find one piece of evidence that the person didn't do it and they are not guilty. But yet in our model, and based on the predictions that we're laying out, the evidence, especially in the realm of function, is on the side of the creationists. And the fact that the purported fusion occurred in the human line, like I said before, would be indirect evidence, because even if there was a fusion event, it can be explained by both sides, especially because we believe in a bottleneck anyways. You guys would have to explain how everybody on this planet supposedly got the fusion, and today all humans got 46 chromosomes, and apes or chimps, for example, have 48. But with the promoter, yeah, like you said, I believe your definition was, where transcription of a gene by RNA polymerase begins. It's kind of like an on switch in the genetics, and that reputed fusion site is overlapped by a functional gene, as you kind of pointed out. So our argument would be that the so-called fusion site, and I would say Tompkins' argument is that it's not a fusion site at all. It's a functional component of that DDX11L2 gene. So this so-called fusion site does lie within the first intron of this gene, but I do find that Ruhif and a lot of the critics of Tompkins do downplay the significance of that, because they say like it's low levels of expression or low levels of function, for example, but that reputed fusion sequence does reveal that it's an active promoter for the transcription of that alternative transcript, but it also binds at least 12 transcription factors. It initiates RNA transcription, which begins within its own sequence, and it has a chromatin profile that further supports its active promoter status. But as I said in my opening, there would have to be a disabled second centromere, also known as a cryptic centromere. And I've had debates on this, and I've asked, the evolutionists to point that so-called disabled centromere out, and I've never had them pointed out. So, maybe you could. But you did mention fusions that have occurred in nature, but the resulting fusions in nature are totally different than the ones that we actually see today, because most germline mutations, the ones that would have to be passed on, this one would have had to occur in the germline versus the somatic lines, of course, do not actually arise as end-to-end fusions, but instead arise due to mistakes in the crossing over process. Like, for example, chromosome fusions can arise within cancer cells end-to-end, but these are not transmitted to the next generation, right? So these crossover fusions result in the deletions of the ends of each fusing chromosome. This means that those special telomere sequences at the ends of both chromosomes should be deleted. And the ones that we do see, these telomeric motifs, they are highly degenerate, and you pointed out that would be expected. But the fact is those so-called repeats are found throughout the genome, and they're actually found to be functional. And one last point too, I'd like to point out is that in most fusions that occur in nature, what's seen is something called satellite DNA. But in this fusion, when they first found it, I believe is in 1991, at the top of my head, it was a fusion that wasn't really expected. It had a different pattern than what is primarily found in living mammals that actually went through a chromosomal 2 fusion. So it didn't actually contain this satellite DNA. And in living mammalian fusions, they find satellite satellite DNA combined with satellite DNA, or satellite DNA combined with telomeric DNA. But telomere to telomere fusions is rarely, if ever, heard of or observed. So this is a different type of fusion, this purported one. Are you familiar with the satellite DNA, and why is this not found in the purported fusion site? So I think it's not just the function that we look to. There's a few discrepancies. Where's this so-called disabled centromere? Where's the satellite DNA? Why is the area so degenerate? You say that the area that's degenerate is expected, but yet these telomere motifs are found throughout the genome and they're functional, which means there's evidence that suggests they're not the remnants of a purported fusion event. So I know I talked a lot there. Take your time, Catz.
Okay, well, I think I've got three questions that you asked about satellite DNA, you asked about the disabled centromere, and you talked about information being lost or telomeres being lost in examples of human. So we'll start with the disabled centromere. Raj, can you put slide 4 on for us, please?
Slide for the title. I have the title here.
Just on the PowerPoint, the fourth. Ignore the numbers on the actual slide itself, just the PowerPoint. No, it's not that one. Just the fourth slide, literally the fourth slide. Have you got PowerPoint?
The fourth slide, babe.
Oh, okay. Literally the fourth.
Yeah, literally, yeah. So when I give those numbers, it's literally just support. Yeah, ignore the numbers.
You can also pin my camera, so you can actually see it in the meets. It's more direct than the actual video.
I wasn't seeing it. I thought I see it on gray.
So essentially, you asked about the disabled centromere. Well, there it is. Highlight that at the beginning. The location has been detected. It's between the bands Q21.3 and Q22.1. It's in that region. That's where it's been detected. So that's the disabled, or the remnants of that disabled centromere. Satellite DNA. Now, I know Tompkins has said, there should be satellite DNA, but satellite DNA are repeating sequences around centromeres, not repeating sequences in telomeres. There's absolutely no reason whatsoever we should have satellite DNA in the telomeric region or where two telomeres are fused, because satellite DNA exists around the centromeres. It doesn't exist in the telomeres. We know that the telomere has that TTA triple G sequence. So I don't know why anyone would expect that to be there in the first place. It just literally shouldn't be there. And the last one you said, Raj, if you can go down to the one that would be numbered 58, the slide that 58, you talked about chromosomes being, some types of fusion deleting like an arm of the chromosome or something like that. We can, there are different types of fusion that can be seen. And like, I don't know if you're familiar with Robert Sounion translocation. I think you probably will be. I know you do a lot of this stuff. But, that's a different type of fusion where it involves a centromere of one chromosome, and that can cause the loss of an arm of the chromosome. That is a different type of fusion to telomere fusion. And I'll not get Raj to keep switching the slide, but I did show you earlier on that there are study after study after study after study after study, so easy to find, we're analysing and seeing in the lab telomere to telomere fusion. Answered all the questions that you had.
I do appreciate that you answered all the questions, yes, and thoroughly too. Unfortunately, I have seen that region before, but that pointed to, I believe it's about 171 base pair repeat with the disabled centromere that characterizes primate centromeres, as in many other places of the human genome, kind of like those telomeric repeats. And this site is way too small and too human to actually be a remnant centromere. And most importantly, this entire sequence is internal to a functional gene as well, just like the reputed fusion site. So this would be impossible if the remnant centromere, that sequence you're pointing to, had ever been a real centromere. And the point of the satellite DNA is all observed fusions, mammalian fusions in nature, do have the presence of this satellite DNA, but yet this purported fusion site is, it would be one of maybe one, or maybe you can point to another one, very rare fusion in nature. And like I said, though, it's occurred in the human line. It is indirect evidence, right? So either way, it would be agnostic. I'm just saying that there is there is certain aspects of it that are inconsistent with it being a fusion site. And Dr. Tompkins has invited people to respond, of course, to his papers that are published in, I believe, the ICR and the Answers Journal. And I know Ruhef, I believe his name is, he's addressed portions of it in like his videos and in his blogs. But there is also limited homology with that region in the chimp genome as well. But I understand that you say, are you saying with the Robertsonian fusion, are you saying that because of that, the remnant telomeric sequences, you would expect them to be kind of small and degenerate the way that we see them? Would that be the reason why you're pointing to that type of fusion?
No, I was pointing to that. Maybe I misunderstood what you said before. I was pointing to that because I thought you'd said that if they had a fused, we'd have had to lose one arm of, we'd have had to lose one arm of the DNA. I'm pretty sure you said we'd lose some DNA somewhere. And I was just pointing out that it's a different type of fusion where we can lose an arm of a chromosome if the centromere is involved in the fusion. That's my point.
And I just say that because I have had people point out that, this purported fusion site was a Robertsonian fusion. Are you claiming that? You're not claiming that, right?
No. It's absolutely not a Robertsonian fusion.
Okay, I'm glad that you said that, because I've had some, proponents fight hard that, supposedly it was. So that's kind of why I said that earlier. But I think the most important part is those internally located telomere repeats that you keep pointing to. I think that's a huge distinguishing factor. Mm-hmm. Those do, just like that area of the disabled centromere that you pointed to with like 171 base pairs, I believe it is. They occur throughout the human genome from only a few repeats in length to literally hundreds of repeats long. So those telomeric repeat sequences are unique to the ends of chromosomes, but then distributed widely over the human genome with various functions. So what you're looking at is possibly the presence of, internally located telomere repeats. Seems like it may not be that actually. So the fact that that area is degenerate and too small, and then you say, well, that's expected. Well, what we do see, that region is throughout the entire genome as functional DNA elements. So I just think there is some genetic discrepancies, but even for sake of argument, if I were to just say, okay, let's just say there was a chromosomal 2 fusion, you would agree, I imagine, that it did occur in the human line after the split between the chimp-human common ancestor. Therefore, both models should theoretically be able to explain it. I think our model could explain it better because we believe in a bottleneck of just 8 people at the flood, which means that it's easy to explain everybody having it. I believe you guys would just kind of say genetic drift, of course, would fixate this fusion in everybody in the population. But at the time of this fusion, when it occurred, how would you say that it was, other than just saying genetic drift, how did everybody on the planet suddenly, like what we see today, everybody has this fusion. So it obviously spread to every single person on the planet. Go ahead.
So the question was, how did it spread to every person on the planet?
Yeah, because I'm just saying if let's just say there was a human chromosome 2 fusion. It's easier to explain from our model because we believe in a bottleneck of only 8 at the flood. So easily that can be passed on to every single person on the planet if there was a fusion in the human line. But for you guys, when did this occur? Because you're just looking at it, kind of like these endogenous retroviruses. You're just looking at the insertions. You've never actually seen them inserted, unless you can correct me on that. You're just kind of inferring. Therefore, how did everybody on the planet, whenever this occurred, how did it get passed on? Why is there nobody with, the 48 chromosomes, for example?
Well, going to the insertions, just a second, you did just say that we don't see ERV insertions happening. An ERV insertion is basically an insertion of genetic material from a virus, isn't it, that kind of like decides it's going to live in your DNA, essentially. We understand it like that, don't we? Right. Well, we use that all the time, We know that viruses are out there. We use them as vectors when we're genetically engineering plants. We use them as vectors when we're genetically engineering bacteria. There's a whole science built around using viruses to introduce DNA. And then when those bacteria, certainly when it's inserted into that plasmid, because they reproduce so quickly, those insertions are reproduced. So there is no doubt that that we can see it. We see it all the time. It just has to obviously happen in a cell that's going to produce a sperm and an egg.
Right. It would have to happen in the germ cell line, you're saying?
Yeah. It would have to happen in that for it to be passed on. But there's no doubt at all. It's a really well understood science using viruses as vectors for genetic modification. Genetic modification built around it.
But these, like endogenous retroviruses, like obviously endogenous retroviruses are just one class of these greater variety of retrotransposons, right? And we can talk about the ALUs, for example. We can talk about the jumping genes. So right now we see in our genetics, we see these so-called viral remnants of ancient viral infections, right, from the hundreds of thousands to millions of years ago. So my question is, and I get what you're saying about, I guess, genetically inducing them, but when we see these insertions in our genetics, in the chimp genetics, in the old world monkey genetics, of course, you're seeing what looks like that, but you didn't actually observe that being inserted, is my point. Because it would have occurred millions of years ago in, say, some Australopithecine or, Neanderthalensis or Homo heidelbergensis. I mean, we don't have the DNA of Erectus or Australopithecus afarensis or Africanus, for example, to determine, hey, this is exactly where this was inserted. That's the whole point of why it's called endogenous. You know what I mean? Because it's ancient. Would you agree with that, at least, before we get into more on that?
Well, I mean, of course, like a lot of things we discussed about, we weren't there when they happened because evolution, it takes a long time. But the point is we can reproduce it and we can see it today like we do with genetic modification. We take viruses, they insert genetic material. That genetic material then becomes part of the host DNA. And with the bacteria, it's easy to see that being passed on because of the binary fusion, the way they reproduce. With the human, it would have to happen in a germline cell. You're right with that. But I don't see any reason why we would have had to be in there. All we have to do is recognize that these insertions have all the characteristics of viral insertions to know, because we do know that viruses do insert genetic material and it can be passed on. All we've got to do is what they are.
Yeah, I think that's a good point. That I would agree that it does have the genetic structure of viruses that we see today, right? What is it? The pol gene and the is it the pol and the gag gene that is kind of significant in this situation? The point I'm making, though, is that so we know, even if we were to just for the audience sake, if we were to just go into viruses briefly, in general, would you agree that we do have trillions of viruses in and on our body? And most of them actually being beneficial to the regulation of the species of bacteria body. But when these viruses cross species, for example, like the coronavirus, it crosses into the human genetics, and now the human genome can't regulate it. It's foreign to it. So therefore, this virus burns hot and fast, and it's of course detrimental to our bodies. But generally, viruses, would you agree, they don't cause disease? They're highly beneficial to the ecosystem, to the bodies, and it's all about regulation, of course. But the problem is, when they jump from a different species, we can see that. We can see them inserting their genetic material, obviously, into our genetics, and then our species don't have the ability to regulate that virus, and that's why they go from good, from wherever they jump from, to bad. So I agree that we do see that. If you want to just kind of touch on the points I made on what you agree with and what you may not.
Yeah, well, I mean, obviously, we do see viruses in certain genetic material, and we do see that genetic material.
Right.
I think, Raj, is it okay to bring up number 28, slide 28 of mine?
Yeah, give me a moment.
Are you saying that we've seen them passed on through the germ cell lines and become an endogenous retrovirus?
What I'm saying is we use them
29?
all the time in genetic modification, when we're genetic modification in plants, in bacteria. We use viruses as a vector. That is how we do it. We use them as a vector to introduce this genetic material, and then we watch that be passed. That a lot of people are against. a genetic modification because of that fact, because we're introducing new genetic material. And you'll see people going ripping crops up in fields, genetically modified organisms. We don't want it because they're worried about how, when that spreads through reproduction, how it's going to affect bigger things. Now, if you're saying it can't be passed on, then all those people are wrong. They're digging up these plants for absolutely nothing. You know what I mean? We know that it's not a case of have we seen it millions of years ago be passed on. It is just a fact. The genetic material, if it's in the germline,
Well, my main point is that no viral load we've ever seen incorporated into a human and passed on today becomes active functioning genetic material. Because the question about these retrotransposons, the ancient ones, not just some that we've seen and genetically modified and seen passed on, the question regarding the all these various classes of retrotransposons that we should be asking, Catz, is are they really the leftover remnants of viral infections in our DNA, or are they created units of DNA function as our model of created heterozygosity would suggest? Because evolutionists have assumed the former, and this is because, as you said, and I agree with everything you said there, they bear strong similarities to actual viruses, and this ultimately should predict that these sequences are non-functional for the most part. But according to our model, we would predict high levels of function in what we would say are DNA elements. But as you probably know, since you understand this topic very well, is that we see the incredibly important functional roles being found in these endogenous retroviruses and in all these other various classes of retrotransposons. So if viral DNA is inserted at random into the genome, as you're saying, it is much more likely to disrupt existing genes. And I think that's what we're seeing with the genetic modification. For example, a blindfolded painter will most likely mess up the painting he is working on by just applying random brushstrokes to it. I use that analogy because of the analogy Pelagia used about because it's not quite like pseudogenes, genetic mistakes, because it is supposedly the insertion of a viral infection. So it's more like a splatter in a book. But that should most likely mess up the painting, and it should mostly mess up the genetics as we see these viruses do. But yet, how would you explain the various roles in determining cell types or regulating genes, for example, important in the embryo and the placenta? How can just a viral insertion suddenly become that highly important in the genetics? Take your time. I know I talked a lot there.
Yeah, no problem. Right, so if we have a look at the structure of the ERV, hopefully I'll answer a couple of questions. The very first thing you said is, address the point that why they're not causing a lot of damage. Well, viruses do cause a lot of damage. Obviously the endogenous ones that have stuck with us have, due to mutation over time, they become inactive. But they've got the recognizable structure. And if we look at the left and the right, we've got the long terminal repeats. Now, this is the reason that a lot of ERVs are accredited with helping regulate function of genes, because a virus, when it inserts its genetic material into our own, it needs our cell to use that genetic material to make more viruses. So it has to be inserting its own We've talked about promoter regions already, right? It needs to insert its own promoter regions, and the long terminal repeats are where we find those promoter regions. So if a viral insertion ends up in our DNA and it becomes inactivated due to mutation or whatever in the GAG pollen, the M section, we've still got those promoter regions there. And, it's not a big leap to then see, well, how they may end up regulating other genes, because they've already got all the equipment to do that. You asked about the various uses that they've got. Now, the vast majority of ERVs we cannot find a use for, but over time, obviously, mutations do happen. And if we look at the ENV gene that you mentioned about placenta, then in animals that have live birth, and only in animals that have live birth with a placenta, the ENV gene enough to create glycoproteins, which are essential to the proper functioning of the placenta. But that doesn't exist in birds, for example, that lay eggs. It doesn't exist. It's a mutation, a beneficial mutation, which has led to an adaptation, which has helped drive evolution, if you like, and that in that one specific section. But unless you can sort of provide to me some citation which shows me that we know the use of a whole load of these ERVs, because I've really looked and I've looked and I've looked and I've looked and I've looked really, really hard. can only find a handful of known uses for ERVs or ERV fragments, and the bulk of them are because of the promoter regions on the long-term repeats.
Well, as you know from your debate with Hovind and just from your videos that I've watched, the full endogenous retroviruses, there's only about 7 or 14 in the human genome, while you got thousands and thousands and thousands of fragments, right? So the ones that are full, these endogenous retroviruses that we can sequence, like say the 7, for example, in humans, those are all highly functional. I mean, I got papers here that says that these ERVs are not only functional, but that they have crucial functions that help regulate genes and even determine cell type. So claiming that they were co-opted, as you just did, and I'm more than aware of that argument. I just think that that's more like uninformative storytelling, because unless you have a paper that can actually show a non-functional endogenous retrovirus going from non-functional to something extremely important in the immune response, in regulating genes, for example, because I've even got another paper here, and I can post them in the chat after. It says ERVs frequently act to distribute regulatory information and thus confer genes with new patterns of expression and function. And I mean, I can go on and on, but for sake of time, I'm not going to. These, even if we just look at the greater class of retrotransposons, the fact that they're extremely good for us, they can, that's why some of them are called jumping genes. They can jump around in the genome, and they can turn on and off various types of genes. And my question to you is, and you kind of touched on it, there's this class of retrotransposons in the mouse embryo that if you deactivate it, Catz, the mouse embryo will develop, but then it just suddenly stops. Well, why? Because it depends on the function of these retrotransposons, which evolution has always assumed was junk DNA. So, but my analogy earlier about the co-option, for example, of these endogenous retroviruses, for example. Like I said before, if viral DNA is inserted at random into the genome, it's much more likely to disrupt existing genes. I think it's sorry to say, I think it's nonsense to say that a mutation led to one of these ancient viral infections to be extremely important in the placenta. What happened with this mouse embryo? If it's required, this class of retrotransposon, did this mouse embryo or this mouse just die every time until it actually got the co-opted function of this retrotransposon? I know I kind of said a lot there. I guess the first question I would want you to ask, do you have any technical papers that can actually show today some viral insertion going from non-functional to functional, or is that just more like inference?
Raj, can you bring up slide 30? Going from, I do have, right.
Excuse me, let me just check with Raj on time.
Yeah, because I know that co-option is like what they say, but I mean, just saying it is not really like an argument. So I'm just, if you did have, like I debated a biologist, for example, and I asked him the same thing. I said, I understand co-option. I understand that these endogenous retroviruses, it's not just about the function, whether they're functional or not. It's that nested hierarchical pattern of distribution that's seen from humans to chimps, to old world monkeys, down to fish, for example, those nested hierarchical patterns. But as I showed in my opening, we too predict those nested hierarchical patterns. Of course, there's going to be more similarities in anatomy, physiology, morphology, and genetics in a human and a chimp than a human and a fish. So that's why if these are created units of DNA function, we predict that they are extremely functional, not just low-level levels of function that we can explain away through co-option, but highly functional. This goes for the pseudogenes, for example. This would go for like the reputed fusion site. And we know ENCODE has revealed that over 80% of the non-coding regions are actively transcribed into RNA suggesting function. So all of this, because like you pointed out, it's all of the factors combined that can tell us which model is more valid, universal ancestry or limited ancestry. Go ahead. If you got the slide up, we can go from there.
So, I mean, just touch on that point there. You seem to be coming from the standing point that ERVs, we've got a whole host of ERVs and they've been extremely, valuable to us. But the fact is that we don't have whole ERV insertions that perform functions. What we have, and we only have, are fractions, small parts of the ERV insertions that have mutated and now can perform a function. So we talked about the EMV mutating to allow placenta formation, etc. But there are no entire ERVs. They are that perform a function as a standalone function. It's small mutated parts. So the question we've got to ask ourselves is, are they evidence for evolution or not? Okay, because we know that these bits of DNA are in our with ours. Now,
I'm sorry to cut you off. I just how many of these? Because think about it. These functions that are found in gene expression, cell differentiation, development, for example, these are found accidentally. Catz. You know, the evolutionists aren't looking for it. It's our prediction that the vast majority are going to be functional. So, how many of these are truly functional if they're accidentally finding them? You know, how many experimental functional tests have been done on not only the endogenous retroviruses, but also all the fragments, the ALUs? Because I know you always talk about the ERVs, but there's a ton of other classes of these fascinating retrotransposons. I call them variation-inducing genetic elements because of the functional roles that they play in cell stress responses and other protective measures. I don't see how these functions can be actually co-opted into the genome. So yeah, my question would be, how many of these experiments have been done on these various classes of retrotransposons to see if they're actually functional, Catz?
So I mean, we could turn that around and we can say how many. What is the evidence that the majority of them are functional at this point? And the evidence is that actually difficult to find a whole load of them that have functions. You know, so it seems a bit more like personal incredulity, you know, just say, well, I think they probably will, you know, have a function. The fact is, are they evidence for evolution, which is what I want to get through on this bit here.
Okay,
now,
go ahead.
So you touched on before and you talked about, I think you said like 7 or 4, and I've made that mistake. I have massively made that mistake in a video where I said we only share 7 with chimps because, I have to correct that, and I didn't do my research properly.
No,
but I did a video.
I would agree with you that even the fragments, the thousands and thousands of fragments, because when I saw you say that, I thought, okay, it's probably just a slip up, because the majority of the fragments, the majority of the 7 that are actually, you know, I guess fully formed and not broken up into these fragments, we do share the majority of them with chimps. I mean, there is a nested hierarchical distribution in all of biological life. I'm not disagreeing with you on that one, if that's what you're getting at. I agree with you on that, 1,000%.
Yeah, but in terms of linking it to evolution, I'll just get through this paper. This one here just looks at one family, okay? And this is comes from a video I did with Paul Odgie lately. He did all the research for it, so I'll thank him for that. But we're looking at the human ERVW, right? We have 211 of these insertions in the human DNA from this one virus. And like you say, you know, it can infect people, it can jump around, etc. We've now got 211 of these in our DNA. Raj, if you can just go on to the next slide. Now, out of these 211 that we've got in our DNA, we share in exactly the same location with chimps, 98 with gorillas, 97% with orangutans, 9% with gibbons, 62% with the rhesus, and, you know, I mean, I've highlighted at the bottom. The 211, 123 of them are in all of them, shared in all the primates. And what we can do with that information, and if you just skip on.
It's a nested hierarchy, right, in the DNA?
Yeah, so if you just skip on to number 33, if you can, Raj. When we trace those insertions back, right, these insertions...
And here's where my problem is, because I looked at this, and I'm sorry to interrupt. I just want to touch on everything. When you say trace them back, this is where the story, this is where the philosophy comes. Because, like I said, in these Australopithecines, Homo erectus, I mean, we do have DNA in you know, the Neanderthalensis and you know, the Denisovans, for example. But a lot of the ones where you've got where you're inferring that these inserted, these are just bones. We don't have the DNA from them. So would you admit that a lot of this chart that you have, it is inference, especially with the fossils. We don't have the DNA to see if this is really where they're inserted. That's why the major question is, are they really the remnants of ancient viral infections, or are they created units of DNA function? And your only argument is co-option, and your other argument is, well, the vast majority probably aren't functional. Yet we know that over 80% of the genome is actively transcribed into RNA. That means there's activity all throughout the majority of the genome. If you could answer that question before you continue.
I will, and I'll just move on to something else afterwards that will help. But no, it's not inference, because if you were to zoom in and you to look at these, you'd see, plenty of these are animals that are alive today. They're animals that we can get,
right?
We can get DNA samples from. It's not inferred at all. But the strength of this,
and those ones are.
Well, hang on. I'm just showing you the one of the ERVs, just one of them.
The AGRV.
We have the ERVW. We have, there's I think 31 families or something like that, and they all support the same phylogenetic tree. They all show the same.
Well, you know that HERVW is functional too, right? I just want you to know that at least.
Right. So, like I said,
that family is active in all tissues except for stomach and I think the prostate. Just going on the top of my head there. So that distribution, that nested hierarchical distribution, I'm just saying it's not really helping the case for universal common descent, because if you want to prove your point, you just chose the wrong example, because that example you just chose is highly functional. So once again, do you have like a paper that can show the co-option of this endogenous retrovirus, this family of endogenous retroviruses going from non-functional to functional? I think that's a really, really important question to be asked or answered.
Can just get back to the question isn't really a basis of function. Like we said before, there are no, and this is where it falls flat for creationism. There are no entire, it's from one long term repeat right through to the next, that serve a function. We have small mutated parts. So when you say the ERVW has a function, it doesn't have a function. There will be small parts of it that have mutated to have a function, and it still is a fact that most of them don't. They are facts. Most of them don't. But where I'm going with this, I really want to get there. Where I'm going with this is, if we build, because we talked about mounting evidence up before, if we take the 31 families of ERVs, they all support the same tree. Now let's have a look at something else that does. Can you go to slide 35 for me, Raj, please?
Will do.
So are you familiar with Apobex? And I'm probably pronounced that wrong. Apobex.
Excuse me, before you get on, do you guys want to go about 50? I only have like one question so far because you guys have been really thorough. Do you want to go for 10, 20 more minutes? I mean, what's good for you? Because I know
I would say
Pat's has something to be.
I think because we're having so much fun here and it's flying by, I can't believe it. We agree to an hour and a half. If you want, we can maybe take some minutes away from the question and answer and just add it.
Yeah, no, absolutely.
Because there's still so much we can discuss. If you want to do
it with you,
if that's okay with you. And I'm just going to let you. I know I kind of talked a lot there and had some clarifying questions. I'm not going to butt in until you're completely done, and then I'll give my thoughts.
Is that good with you, Catz? About 10, 20 more minutes.
That's great. I'll only need about 5 on this, but that's brilliant. Yeah. Thanks.
Okay,
also I just checked the spelling, because my open office is not seeing this word. It came up with apogees. That's not the word I'm looking for.
No, that's not the word you're looking for.
No.
Okay, go ahead.
So the point I'm going to make here is that it's the sum of all the genetic evidence. Now, the sheer, going back to just before I touched on the Apobex families we've got of ERVs, the sheer number of insertions which happen at completely random places, to have them in exactly the same position 98% of the time with our closest relatives, as predicted. And the thing is, we are sharing them with the closest relatives that have been predicted by fossil record. And we already had that tree before genetics come along, and the trees allowed us to make a prediction about what the ERVs would say. The ERVs are backing that up. Now, another thing that backs that up are this. It's a family of, you can see on the screen there, but if we go to slide, and can you do that, Raj, please? Just 2 more down.
Which one? You broke up there.
37, please. Now,
there we are.
the Apobex are a family of genes that code for, it says they're like deaminase enzymes, and they've got something to do with hydrolyzing amino acids. And they're what we call like a zinc-dependent deaminase. So I'm assuming, I'm not a biochemist, but they somehow attack some, a zinc bond in an amino acid or whatever. I don't know. But they are involved in hydrolyzing amino acids. That's the function. And when we look at the tree that we built backwards, it does appear that we can't find the genes for these. If you just skip on to the next slide for me, please, Raj. We can't find these apebex vertebrates. We can only find them in vertebrates. We can't find them in other animals. And that would allow us to make yet another prediction. And that prediction would be, if we only find them in there, then the mutation that's caused that, and they are similar to all you've seen on the screen there, are other types of zinc-dependent deaminase enzymes like that, okay? That whatever mutation caused that was caused when the vertebrate line was splitting off. So how does that fit in with the phylogenetic tree that we have already created through the use of other techniques, other genetic techniques, other dating techniques, and fossil records? If you skip on to the next slide, please, Raj. Well, what we see is this, and what we've got on the right-hand side is one of those phylogenetic trees. Now, I'll point out, because it's too small on the screen, in the top right-hand corner, these are there's like a color code. These are the different versions of these apebec genes, the different types that we've got. And if you look at that phylogenetic tree, it looks like a graph coming out, and that graph shows us a color code of how frequent species each of those genes are. Now, if we go on to the next slide, Raj, number 40. If we were to then...
By the way, Catz, if you need to have me zoom in on something, I can zoom in on it if you want me to.
Oh, thank you.
Ooh, he's giving you all the stuff.
If we were then... Oh, sorry. If we were then to zoom into specific parts of the tree, what the prediction evolution would make is, within species that we've already determined are closely related, already, before we've even done this analysis, we should see extremely similar distributions of these, the frequencies of each of these types of apebec gene. And we can look at primates there, and we can look at carnivores, and we can clearly see that the primates and the carnivores are different to each other, but within the primates and within the carnivores, they show exactly what evolution would predict. If you nip onto 41, the next, so the one with the marsupials on. So hopefully that's on the screen now. The marsupial one, can you see that? Yeah.
Yeah, I'm zooming. I'm getting it there. There it is.
Okay, so we've got again another group. We've got the marsupials that we will predict would all be absolutely bob on, so close to each other, and then we have the rodentia below. Again, they are different to the marsupials, but they are very, very similar to each other. And all of this is a prediction of evolution based on genes, which also matches the prediction used based on with ERVs, which also matches the fossil record, etc. And so you see how the pattern builds up.
Awesome. I appreciate that. Lots of points there. I'm going to try and unpack it the best I can. So it seems certain genes require certain elements. This is just an internal nested hierarchy, as I said, we should both expect. We would expect land mammals to require certain elements more than aquatic life. It just comes down to the agnostic part of the model, because it all comes down to where does genetic diversity come from, for example. And the big difference, and I talked a lot about this in my opening, so if you're listening, you'll understand what I'm saying. The big difference between my view and then your view is that I explain the vast majority of nuclear DNA differences not by mutation. I don't explain the vast majority of our genome by mutation and evolutionary leftovers and ancient viral infections. This is how evolutionists explain them. But we explain them by pre-existing or created diversity, right? This is the created heterozygosity hypothesis. So this one genetic difference, Catz, has massive implications for the timing as to the origin of species, the function of the genetics of these various species. Like a lot of evolutionists will say, how do you get all these species, from a few archetypes after just a few thousand years, and it's like, well, if you're trying to explain the origin of species just by mutations, then yeah, of course you're going to need millions of years for all these differences to accumulate. But if the differences are there from the start, God encoded Adam and Eve, and this applies universally to the kinds, with pre-existing functional DNA differences from the start, the simple operation of recombination and gene conversion and other processes, of course, inbreeding and migration and isolation, can produce new species and visible variety quickly. But the point of that is our model predicts that the vast majority of the genome and these DNA elements that we're talking about, specifically the ERVs and other classes of retrotransposons, ALUs I really like as well, because there's a lot of papers showing how functional and important those are to the genomes. That's a head-to-head prediction. Is the vast majority of our genome based on these genomic fossils and evolutionary leftovers, or is it more consistent with our prediction of created diversity? I find it funny because evolutionists, and I think you're proving this for me, they seem to think to themselves that if we are really made in the image of God, then shouldn't we as humans be absolutely distinct from the rest of biology? The Bible doesn't imply in any way that we have biological life and then all of a sudden we have humans, which has absolutely no similarity to any other species out there. Like, there's no reason for us to believe that. There's no reason that even, you should believe that we shouldn't be more similar to one species, for example, the apes, than to another species, for example, a wolf. And we can see if we start from the Genesis starting point, God tells us he created us in his image. So therefore, we can look at humans and how they've made things, designed things, for example. This can give us an idea or an inspiration into the patterns in which God may have used to ultimately create life, a biological life in the beginning. And it just so happens that we build and design modes of transportation, for example, in groups within groups, nested hierarchical patterns. So yeah, we're going to see a nested hierarchy in DNA. We're going to see a nested hierarchical distribution of these DNA elements, these endogenous retroviruses, from humans to chimps to the old world monkeys to fish, and so on and so forth. That's agnostic. We both predict that, and I think you would even agree that, of course, our model would say we're going to share more with a chimp than we do with the fish. It's a groups within groups patterns of life. But our predictions are the ones that are coming true. I mean, the ENCODE project is revealing that most of the genome is actively transcribed into RNA. We've made specific predictions. You talked about predictions at the beginning. Dr. Nathaniel Jensen has predicted that based on mutagenesis, for example, knockout experiments, we can discover just how active it is, because that experiment, or that the ENCODE project, they define function as biochemical activity. So we know it's active, but exactly what it's doing, what are the rest of these endogenous retroviruses doing, other than the ones that we just accidentally found have important functional roles? And I understand you say mutations, for example, can make certain parts of them, like that protein syncytin, like functional in the placenta. But like I said, I'm just saying that that's more the evolution philosophy. My question to you would be, if the majority of these DNA elements, let's say the endogenous retroviruses, or just the majority of the non-coding region, were truly junk. Simple question. Why would the cell bother with it? Because such an activity, Catz, would be wasteful of energy and resources and would have been eliminated by natural selection long ago. But the fact is, we now know that over 80% of the genome is actively transcribed into RNA, and these RNA elements are incredibly important in the expression, the regulation. So yeah, how would you answer? Because your argument seems to be, well, the majority of them are still shown to be, functionless, for example. So yeah, how would you answer that question?
Well, the majority of ERVs are shown to be functionless because we haven't. There's no evidence, apart from the few that we've managed to find the function for, that they have a function.
I just want to go on record and say, I'm saying there's a lot more than just a few that are functional, and these were found accidentally. My question is just pretty straightforward. Like, if it's all still truly junk, the genetics, why would the cell bother with it? Because such an activity would be wasteful of energy and resources, and natural selection, of course, should have eliminated all this useless evolutionary leftovers long ago. We can just look at conserved genes. We can see that mice have conserved gene sequences that are also the same in humans. Evolutionists will say, okay, that shows that they must be functional, because if they were truly junk, then natural selection is obviously not going to preserve those gene sequences, and they're going to accumulate mutations and they're going to diverge in time. So if mice and humans are obviously further on the tree of life than humans and chimps. So we would, like, for example, the cytochrome C, which we can get into, is more similar in chimps and humans than it is in humans and mice. So yeah, that's my question. Like, why would the cell bother with it? Because I'm saying the vast majority of our genome is functional. That's a prediction from our model. You're saying it's not, and the vast majority of these ERVs are not. So then why is the cell even bothering with all of this useless baggage? Go ahead, take your time.
So, well, first of all, I mean, there is some non-coding DNA which is needed for sort of like the structural purposes for wrapping the DNA into the nucleosomes and around the histones, etc., in the chromosome. I can't remember too much of what I did about the structural DNA, so maybe I might have used one or two of the wrong terms, but a lot of the junk DNA is involved for structural reasons. But secondly, what was your other point? You had one other point that you wanted. Oh, yeah. Sorry. No, I had another thing.
Yeah, if it were truly junk, the vast majority of the genome, why would the cell bother with it is technically the question I have.
I think why would it? If it was creation, why would it be like it is? But if it was evolution, where we're having insertions, we're having jumping genes, we're having mutations, we're having DNA and genes moving from one place to another. I showed you the ERVs before, and I said with that one ERV, just that one, 211 insertions. Now, if that one ERV does a good job, why do we need 211 of it? Why do we need that? Why do we need different frequencies of different genes? Where that doesn't seem to make sense. If it was all perfect design,
I did explain that, but yeah, no, a lot of those are fragments. You're pointing out too.
Oh, but we do have multiple copies of genes. I mean, that's not.
Yeah, that's why they're part of a bigger class of repetitive DNA sequences, right? I mean, the majority of a lot of the genome is redundant, and yet this backup code, right? Those third position codons, they're not just there to tolerate error. Now we know that they help slow and speed processes in the cell. The information compression on every single nucleotide is actually mind-boggling. And like I said, you're saying, if it was creation, why would our genetics look like this? But I'm telling you that it doesn't look the way that evolution has predicted, since we know that there's evidence that over 80% of it is active. Okay, what exactly does every single sequence and every single DNA element do? Well, like I said, we've made specific predictions. You said creationists don't make predictions, but like we do. I can send you paper after paper where we're looking at specific sequences, and we're saying, based on mutagenesis or based on knockout experiments, these sections of DNA that evolutionists are saying are neutral junk may actually do this. And then therefore these nested hierarchical patterns fit better in our model. Because like everything you're saying with the distribution of these retrotransposons and the nice nested hierarchical patterns, I believe that if true, if it's all just truly junk and it's all based on, for example, the cytochrome C, you would show nested hierarchical patterns and distribution of that across all of biological life. And those small differences, you would say, are just a measure of time, divergence time, since all the other common ancestors, of course. And then I would say, like, for example, Hovind, so common design. But your question was, well, then why didn't God just make that cytochrome C gene? Why didn't you just make it the same for all the organisms? Why does it look like there's a nested hierarchical pattern in all of life? DNA, physiology, anatomy, morphology. I agree with you on that. These are the agnostic parts of the discussion. I really do believe it comes down to function versus non-function. And if I can prove that the vast majority of our genetics is functional, specific to the organisms too, like if we're going to determine the kind boundaries, for example, then those patterns become evidence for design and not evolution from common descent. Does any of that make sense, Catz? Yeah. And if not,
so I don't want to put words in your mouth, but what I'm hearing from you, I could show you the strongest evidence based on DNA that that these patterns are produced time and time again, no matter what genetic markers, no matter what genetic evidence I show you. I could show you all of that, but I'm guessing that you would say, I still agree with that, but. And that's kind of what I'm feeling. And you said something else that I just need to. What was the other thing you said just briefly? What did you say?
I was just saying that well, obviously we know there's inconsistencies with the tree of life, right? We can talk all day about convergent evolution. We can talk about loss of traits evolution. We can talk about incomplete lineage sorting. We can talk about these taxonomically restricted and essential genes called orphan genes that break the phylogeny as well. But overall, the rule is there is a nested pattern, this groups within groups pattern that we see in life. So yeah, there's some inconsistencies. You could probably point to some inconsistencies in my design modes of transportation, nested hierarchies, right? But the rule is, yeah, there is these patterns that exist, but I'm saying these patterns exist for functional purposes. But if you could show me that these patterns don't exist for functional purposes, then I would concede that, yes, this looks like evolution by universal common descent. And that's why we make specific predictions. Go ahead. I've talked a lot. You take as much time as you need.
Say then that it seems to me as if you're saying that the genetic evidence doesn't in any way contradict evolution. I would say that you're happy, and if I'm putting words in your mouth, tell me, but I would say that you're happy to admit that there's nothing about genetics and the way that the genetics plays out that in any way contradicts evolution. What you're saying is you think that we've got more to learn about the genome, and when we find out that stuff about the genome, that will add more evidence to creationism. Am I right or wrong on that?
You're partially right, but when you say like when we find out, I'm saying we've already discovered that over 80% of the genome is active to some extent. But obviously a lot of these pseudo genes, for example, we know that a lot of them play important roles in healthy cell processes in the cell, of course, in immune responses as well. The endogenous retroviruses, the ALUs show, function. But like you said, not all of them have function, but we're accidentally finding these functional roles. So we're predicting that these are created units of DNA function. Evolutionists like to look at nested hierarchical patterns in DNA, in anatomy, and morphology. And I just point out, like, we expect the same thing. Of course, we're going to share more of these DNA elements with chimps than we do with the old world monkeys, than we do with the fish. Like, these nested patterns are agnostic to the debate. That's why I'm saying, yeah, if you could show that the vast majority of our genetics, for example, is the result of descent with modification, then yeah, I would have to concede, this looks like, banana plants and whales are related through common ancestry. But I'm saying that the genetic data right now, and we didn't even touch on mitochondrial Eve and Y chromosome Adam, for example, and the low genetic diversity that points us to two common ancestors just thousands of years. We don't even touch on all that. We could do that in a second debate. Yeah, go ahead. Take your time. So you were kind of right.
I think, yes. So, yeah, I think essentially then, what I would need, because you've kind of told me that really the debate that we're having now almost is kind of like secondary, because you don't disagree that genetics supports evolution. You don't disagree with it. So I could show you all the stuff and you would say, yeah, I agree with that. But then you're also telling me other things that, you're telling me that the entirety of our DNA, for example, is kind of like almost perfect without fault. It's designed, every bit's got a little job to do. And what I would need for that is, because to me, that still wouldn't rule out evolution. But I'd need citations, I need papers, I need studies, and I've tried to bring as many as I can tonight. Maybe next time, because I'd like to do this again. Maybe you could bring some next time. But to me, that would be pretty useful, because it does seem that whatever I give you're going to agree with anyway. You're going to agree that cytochrome C is odd. Why did God create so many different versions of the same gene? You're going to agree with that.
That's a good question.
So we're not. Even if I give you all the best sort of evidence, you're going to say, yeah, I agree with it, but I still think God did it. I think that's where we are.
Well, I do have a ton of papers here that I'm referencing for these various functional roles in, like, for example, pseudogenes, endogenous retroviruses, ALUs. So I'll send those, or I'll just post them in the chat, actually.
Okay.
Yeah, and the thing is our model of created heterozygosity would predict that many of these species are forming from what's called shifts in heterozygosity to homozygosity. And this is actually what we see in most speciation events. Like I think it was last year, we seen a new species of finch produced on the Galapagos Islands. So even the changes we see, not to mention, like one of the questions I had, and I know the debate's probably coming to an end quickly, we could talk about cytochrome C, we could talk about so much, but I do want to ask you, like, if we know that we're accumulating, 100 new mutations per person per generation, okay, and we know that the vast majority of our genome has evidence for function, that means most of those are actually deleterious to our genome, and the fact that they're nearly neutral, selection can't do anything about them. So if we track that back to a point of least genetic entropy, least genetic damage, that would actually be a point of creation, a point of perfect genetics. Like how would you, like, if evolution is true, how do you, what type of selection can rid the population of all these deleterious mutations that are actually degrading our information system?
Okay, can we wrap it up?
Yeah, take as much time as you need to answer that question.
Catz, after Catz is done, and then we go
I'll
into
give him the last word, yeah.
Yeah, thank you, hon.
Very quick on this point then. Raj, can you just get a slide number 43 up, please, mate?
Will do. Let me transition. There we go.
All right, so slide number 43, we're talking about mutation rates. There's a paper reference by Cole et al. I should have got the proper heading for it. And, if we look at bacteria and we're looking at the fact we know bacteria can reproduce in 15, 16 minutes. They do, the number of generations that bacteria will have reproduced over compared to humans is absolutely enormous. And we can see in that paper there that we do see genetic decay. That is a real thing. We see it. But yeah, even though bacteria have been around as long as humans have, right, on your Earth, they have multiplied thousands and thousands and thousands and thousands and thousands and thousands of times more than humans have. And they are still going strong despite this decay. And this is what I meant before. This is when I was talking about, this is why we have these bits of junk DNA. This is why we have this sort of like what you think is an imperfect genome. It happens, and life gets on with it. That's evolution. The strongest survive. It impacts as we die out. We see that in bacteria. We compare that to humans. We would have to wait millennia until humans have caught up with bacteria for the amount of genetic decay. Bacteria are still going strong. So it's not an issue.
Did you want to just go right into concluding statements, really short ones? I can maybe address a couple of those points there and then call it a day there, Marvel Girl.
I just have 2 questions, both for Standing for Truth,
Sure.
From the chat, and then the super chats, and to say thanks. Yeah, so that's great if you want to do that.
Sure. Yeah, I'll do just a brief, I guess, just my closing. I want to say that it was, and I know we agreed to 5-minute closings. I'll only need a minute here. I want to say thanks to Catz for doing this. I actually really enjoyed this. I thought it was a highly, scientific discussion with somebody who understands the evidence like yourself. I really enjoyed this debate. Actually, probably one of the best that I've had. So, and thanks for setting it up, a Marvel Girl. I would say that in regards to genetic entropy, we do. I think you kind of dodged the question because you didn't really give a type of selection that, like, there's these rescue mechanisms that population geneticists look to. They're called, they're artificially contrived. They're called synergistic epistasis and mutation count mechanism, for example, to explain the degeneration. Like they recognize that it's a real problem. And we can see that it's a pandemic. It's literally entropic degeneration on all levels. I mean, Alzheimer's disease, a lot of this is environmental and epigenetic, but you see dementia, Alzheimer's, autism, asthma, autoimmune diseases, cancers, immunological diseases. We can see it in the fossil record. We know that the Homo floresiensis, Heidelbergensis, Naledi, Erectus, Neanderthal, they all have evidence for genomic decay. I've got a paper here suggesting that mammoth populations were highly inbred, carried an elevated genetic load. Those so-called pre-humans that evolutionists look to as our so-called ancestors, they're actually just modern humans that were isolated post-Babel and became highly degenerate. And that explains a lot of their features. For example, Homo naledi. You talked about bacteria. Well, we can look at the long-term Lenski experiment. What do we see? We see reductive evolution. All the beneficial mutations are reductive, loss of function, loss of promoter, loss of genes. Even bacteria in nature, they're prone to what's called reductive evolution. You can literally just Google it, and paper upon papers. So even in this Lenski experiment, they're getting rid of genes short-term, but it's long-term degeneration. Lenski, who's trying to prove some type of large-scale evolution, has revealed clear evidence of genetic degeneration. I love how you talk about, how many generations, how fast these bacteria produce, and all we've ever seen is bacteria produces bacteria. But the evolutionist wants us to believe that a bacteria-like organism evolved into a whale over billions and billions of years. But the genetic data just suggests that can't be true. The nested hierarchical patterns in anatomy, morphology, DNA, and physiology is better understood as design-related because of the incredible amounts of function that we see in the genetics. So, I mean, I can go on and on. I'll end it there. I want to say thanks again.
Thank you.
Really good discussion, and I'd be open to doing another one at any time. So I'll give Catz the last words for his concluding statement.
So standing for truth, just one thing. I was messing around a bit. So that was a long minute. Just saying. I added a countdown counter there.
So however long I took, give Catz the same amount.
Yeah, sure. No, it was just I was just taking the piss with the countdown thing.
We're just having a good time, huh?
Yeah.
It was a good time. It was a good time.
I'll only need less. I will genuinely just need less than a minute because I have enjoyed it. I have enjoyed the discussion. I really have. I've enjoyed all the things I've had to research doing it. But it does seem, like I said before, that we pretty much agree that the genetics doesn't in any way, shape or form defy evolution, and the genetics is pretty much what we'd expect to see if evolution was true. However, I understand standing for truth's point is the fact that, well, God made it that way. My viewpoint will be, well, God can do whatever, if there is a God, he can do whatever he wants. So why couldn't he have created life through evolution? Why couldn't he have done it that way? So I don't think we're going to get a winner on the battleground of genetics if he basically agrees with what I say about genetics anyway. If he agrees with the questions about cytochrome C, if he agrees with, the nested hierarchies we get from genetic markers. So we need another battleground, which might be radioactive dating, maybe. The Earth is over 6,000 years old, radiometric dating. And I'd love to do it again on that topic, maybe.
Great.
Exactly a minute. Awesome.
A teacher would know, right?
Yeah.
So let me do the questions. There's 3. I just got another one. Standing for Truth from Area 85 Restorations with a $10 super chat. So they really want to know the answer to this.
Nice. I'll make it worthy of the $10.
Thank you. I would appreciate it on their behalf. Can the creationism model relating to genetics make future novel predictions? If not, nothing you have said during this debate, the entire debate, means anything because evolution has made these predictions.
Oh man, such a good question. I could probably go on for 3 hours about the predictions.
Oh no!
I'm not gonna make it.
Oh, there's another.
I'm gonna make it short. I mean, I think they can easily tell that based on our model of created heterozygosity, we would predict that the vast majority of our genetics and DNA elements are functional. I mean, we literally spent an hour discussing that. That is a novel future prediction that are on paper. So I mean, I could show those after. Also, we make predictions based on DNA barcoding and orphan genes and taxonomically restricted endogenous retroviruses as a way to determine where that boundary is. People like R and Ra, they'll say, where does the phylogeny break down? Well, guess what? We all predict nested hierarchical patterns. Therefore, we need to look to genetics and make these predictions. Let's say we say family level is the kind boundary. So we're going to predict, certain functional roles, like in the cytochrome C gene, for example, that are specific to these species, to these creatures. That's why there's a little known phenomenon called protein moonlighting. These proteins are multifunctional. So that's what we're predicting about the ATP synthase proteins as well. That'll break the hierarchy that Catz looks to in the cytochrome C, for example. We can really dig deep in the mitochondria and look at the CO1 gene in the mitochondria, and that's really conserved. And we can look at barcoding, and we can figure out a way to determine what's related and what's not. But one really specific prediction, and I'm done. Dr. Nathaniel Jensen, because he has papers on the Y chromosome, the mitochondrial DNA, that takes us right back to 2 ancestors just thousands of years ago, mitochondrial Eve, Y chromosome Adam. So the evolutionists have a problem with that. So he said, okay. He looked at African people groups, for example, the Khoisan peoples, and he said, I'll show you how accurate the mutation rate is, showing that Eve lived just thousands of years ago. I predict the mutation rate in these Khoisan peoples. And he said evolutionists can do the same thing. And they admitted, there's a 4-hour debate between him and an evolutionist. He admitted that's a tough thing to do. But he made that direct prediction that only future observations can tell us if it's true or not. So I challenge the evolutionists to go make those same predictions. And that's just a few of probably the hundreds I can go over. So really good question.
Wow.
I appreciate it.
Yeah, great question, great answer. One just came in as well with a $5 super chat from Snake Was Right. I believe probably Pliskin is what you're talking about. Standing for truth again. How can you say synergistic epitasis? Did I say that right?
Oh, yeah,
Thank you.
Epistasis.
I'm close. Simultaneous causes fitness decreases while also saying it isn't visible to selection.
So good question. I've had many debates with Snake Was Right. I think he's in school for cell biology. I'd recommend going to watch those. Yeah, he's fun to talk to. Lots of discussions. You can watch those. We've touched on all these points. Synergistic epistasis is the artificially contrived mechanism that population geneticists are kind of looking to solve the degeneration problem. But to the extent that there is a substantial amount of synergistic epistasis happening, it actually makes the genetic situation worse because what it does is it'll amplify the deleterious effects of mutations, and then eventually, Mother Nature selection will be able to see these mutations. But all it does is it leads to further extinction in certain populations. But overall, the bigger picture is these 100 new mutations per person per generation are accumulating, and the majority of beneficial mutations are going to escape positive selection. The few rare beneficials, they may have a high impact. Natural selection can amplify the best beneficial mutations, sickle cell anemia, for example, melanoprotein mutate. Actually, a lot of these are shown to be epigenetic regulations now, actually. But it's those vast majority of invisible mutations, the nearly neutral ones, those are the ones that build up in our genetics and slowly degenerated us. And you've seen in my closing, I went over a few lines of evidence showing the degeneration process at work and showing that it has been at work in the past. But I have a really extensive paper on synergistic epistasis written by somebody a lot smarter than I am, Dr. John Sanford. So I'll post that in the comment section as well for everybody to see.
Okay, so Tracted has a couple. I'll just take the one I see right here. So Adam and Eve were created with pre-existing DNA differences, including different endogenous retroviruses?
Good questions. Yeah. So as I was saying earlier, these DNA elements like the transposable elements, the endogenous retroviruses, because of their functional roles in, so much determining cell types, their important roles in cell stress responses. Literally, many of these retrotransposons, and I can go on and on. I'm fascinated by this topic, but they have the start sequence for many different genes, especially genes in the brain. And these jumping genes can jump around the genome, turning on and off genes, performing functional roles. Like, we're just beginning to understand the level of genetic multifunctional. The genome is nested, and as far as I'm concerned, regarding the knockout test, the test done based on mutagenesis, we understand less than 1% of the genome. So yeah, these ERVs were created as variation-inducing genetic elements at the start of creation based on that created genetic heterozygosity hypothesis. That's why, obviously, even an evolutionist should be able to say, okay, if that's true, one way to determine if that hypothesis has any validity to it, we need to study the genetics and see if the vast majority of these created units are functional. So I think that's pretty straightforward. Good question. Thanks.
Okay, last one. Standing for truth again. From our friend Peter Quinn. How do you explain the Habsburg?
Oh, you lost me there. Can you ask that question again?
How do you explain the Habsburg?
The Habsburg? Does that have to do with like?
I don't know. If you don't know, I don't know. Does anybody know?
I'll have to claim it.
They might mean the Habsburgs dynasty. They were inbred, I think.
Oh.
I think that's maybe what they mean.
I would say I always point to inbreeding as a way to show that the evolutionary out of Africa scenario is literally impossible. I mean, there's this near extinction event that reduces the population 70,000 years ago to 2 to 10,000. And for many, many generations, they inbred. Look at the cheetahs today. We got 6,000 that are going to go extinct because of inbreeding and genetic damage. So inbreeding, we would predict. Post-Babel, that's how we explain all these so-called pre-humans, Neanderthal, hobbits, Heidelbergensis, erectus, Naledi, for example.
I'm sorry, I'm sorry, just to clarify, Habsburg royal family, and I think Cliff was right. Thank you, Cliff.
Yeah, no problem.
Is that in the, is that like, what is that? Is that Y chromosome? Are they looking at a specific, I'm not too sure.
I guess it was, well,
if it is inbreeding, yeah,
if
he wants to be a little more thorough.
Yeah, I'll send him, you know what, he can send it to me, Peter, if you want to give me more, of a, outlined question, I can send it on to him in email, okay? So, yeah, or you can do this again next time. I want to do the super chats, but I want to also thank you guys so much. This has been great. Before, just in case anybody has to leave before I finish, this has been wonderful, and I thank you both. From the bottom of my heart.
I had fun talking to Catz. I want to thank him. I've been looking forward to this one after watching his debate with Hovind, so I appreciate you doing it.
No, I've really enjoyed it. It's been a lot better to speak to someone who at least understands a bit of terminology. So
Are you
prepared?
Are you?
I
knew you were gonna say it. Well, you win this time, Marvel Girl.
This one time.
I appreciate Cat's sense of humor.
He's great. Yeah.
Did you hear the song, by the way?
Yes.
Of course.
Can we play it on the outro, Raj? Can you do that?
I'll have to change. Yeah, making me do work and shit.
Maybe before the outro. Before the outro, just add it maybe somehow. Thank you.
Yeah, I can throw it in the window.
Hey, Mark, are we gonna read off the rest of the super chat?
Yes, we are reading off the super chats. Okay. I'm going to start with mistake not with 10 pounds. That's pounds, right? Yeah. Argyle says hello. Thank you. Hi, Argyle. Go ahead, Cliff, you gonna do the next one?
Yeah. So Crafty Keela for €2. The Catz wins. Debate over. And that's a raffle emoji.
Now I'm gonna cry myself to sleep. Your chat's so mean.
Again, Crafty with €5. The genetics claim that every human stems from Adam and Eve makes as much sense as homeopathy does to medicine. Ooh.
Hey, I mean, I gave a number of lines of evidence, including Y chromosome variation. Actually, you should look into the 3 major haplogroups that in mitochondrial DNA that take us right back to Noah's 3 daughters-in-law. I mean, genetics, like I said, rocks, fossils, geography, these things aren't passed on sperm and egg. They're indirect lines of evidence in determining ancestry. I believe in limited ancestry. Catz believes in a universal common ancestry. And the genetics, we can trace it right back to 2 ancestors, Y chromosome Adam and a mitochondrial Eve. And these are based on the empirical method. We're not using phylogenetic evolutionary assumptions to come up with the dates. And these are just the facts. It's fascinating.
I would follow that up though with another one from Subtracted, and I'd like both of you to ring in on this if you would. How do you then explain the population growth?
Yeah, that's a good question. So
from Adam and Eve to now.
Actually, that's a really good question because, I actually touched on it. So there's something called the HapMap project, and it actually showed that the genome is young simply based on the chromosomes that we have in the genetic structure in our DNA. For example, we have a lot of large linkage groups still, but every generation we know we experience rounds of recombination, gene conversion. So deep time evolution would suggest that these should all be scrambled to randomness, but yet, these large linkage groups still exist. And there's people like Dr. Robert Carter and Dr. John Sanford that can trace these back to the original sequences. So, that's really fascinating work as well. And the population growth, for example, this new paper on the Y chromosome that suggests the Y chromosome based on high sequence gene sequencing for example, actually only goes back based on mutations 4,500 years ago to Y chromosome Noah. So he's made predictions based on population growth in not only the Y chromosome, but also the mitochondrial DNA, that if this is true, if our human species only go back 6,000 years ago, we should be able to read the history of civilization off of our DNA. The transatlantic slave trade, for example, or the Roman Empire, the Greek Empire, the Mongol conquest, for example. So some of those predictions have actually come true. I can put those papers as well. And that all has to do with population growth and the history of humanity. So more fascinating work. Catz, you can go ahead.
So what was the question about the growth in population?
Yeah, how we can be sure that there's we know that we have data saying, all right, at this point we had this many people. So it can't be from Adam and Eve. I don't want to actually say anything because I'm not in a debate.
Yeah, the growth does go back to Adam and Eve.
The closest I can come to that is we can use mitochondrial DNA, and we can use the Y chromosome to trace the movement out of Africa. Mitochondrial DNA, because the mutation rate is so high, it becomes ideal that when we find early humans and we can test the DNA, we can almost like put the mitochondrial DNA mutations next to each other, like you would with fossils, to go from like the most similar to the least similar. We can trace a path, and we can do similar things with the Y chromosome, and we can show the paths that humans took to migrate as they come out of Africa. But the interesting thing is, and I had a paper on this, but we never got to it. The interesting thing is when you actually trace and you age them from the mutation rates expected for Y chromosomes and for mitochondrial DNA, it actually matches up really, really well with the archaeological record that you get as well. I'm waffling now.
No, that's wonderful. Thank you. So next one, Cliff, it's your turn, I believe.
Yep. So Anthony Purcell with $5 throwing some shade your way. He says, Standing for Truth failed to present any argument beyond a series of unsubstantiated assertions.
He's coming at me. Well,
he is.
Like I said, there's a lot of reasons for myself to cry tonight to sleep. But you know what? Just rewatch the opening. I did, and I'm glad that Catz touched on the Y chromosome variation and mitochondrial DNA as well. That's why I talked about based on high sequence genetics, for example, on the Y chromosome. It shows, and I talked about it in my opening as well, that the Y chromosome variation and the mutations in the Y chromosome only go back about 4,500 years to Y chromosome Noah. And then the mitochondrial DNA, simply based on the empirically observed mutation rate, studies show that mitochondrial Eve lived just recently, thousands of years ago. So the evolutionists, like Catz said, they have done similar analyses, but rather, and he said it there with archaeology, it's fascinating, the evolutionists don't realize it. Rather than using the actually observed mutation rates, they're compelled to use hypothetical mutation rates that are about 20-fold lower than what is actually observed. Mutation rates across the board in the mitochondrial DNA are high. And then they justify this, just like Catz did, based on certain evolutionary assumptions on the geological column, assumptions in archaeology. So we just use the empirical method, and it takes our ancestors, Adam and Eve, just back 6,000 years ago for Eve, 4,500 years ago for...
But isn't that different from the biblical
Noah.
Adam and Eve? That's totally different.
Right, it would be based on worldviews, but yeah, the evolutionists will say that Adam and Eve only go back, they go back 200,000 years ago, for example, and they weren't the Adam and Eve of the Bible. But like I said, based on the empirical method, based on predictions on the history of humanity and the Y chromosome and the mitochondrial DNA, they go back just thousands of years ago. The out of Africa theory has been destroyed.
So may I ask if you're a Young Earth Creationist?
Yes, yeah.
You are Young Earth. Okay, that's interesting.
I hope I made that clear in my opening. But yeah, good question.
I just want to make sure, because people had asked. And great, thank you.
I just have to, unfortunately, because I put my kids down for a nap, and I agreed with my wife that we do the debate at 4:30.
Absolutely.
I want you to, if you have super chats, of course, read them off real quick.
I will absolutely do that real quick.
With answering.
I'm sure you both are. Real quick, let's go in from Anthony again for $5 Australian, invoking an oh my God, I can't speak it. Omnipotent. I know the word. I'm Catholic. Deity. You can explain anything, but you can't make any predictions against which observations can be compared. Thank you, Anthony.
Yeah, well, I'll just say one thing. It's funny because it's like the evolution. It's like it goes in one ear and out the other. I'm not saying that's with Catz. I think he's understanding a lot of what I'm saying. We have a Dr. Nathaniel Jensen. He literally pointed at African people groups where their measured mutation rates in the mitochondrial DNA have not been measured. They're not known. And he literally pointed at them and said, this is what I predict based on genetic diversity in African people groups and non-African people groups and mutation rates in non-African people groups that we know. He made a prediction. It's literally on paper. This is how many times a generation they're going to mutate. Now, all they need to do is get some blood and some DNA from these people groups, and let's see if the prediction's true. Like, that is a direct prediction. That's like me pointing at somebody on the street and saying, hey, you ever played baseball before? And they say, yeah, of course. I say, you're going to hit a ball this far. Like, that is as precise and specific. I just don't like when the evolutionists straw man and or misrepresent the model, because whether you agree with it or not, there are predictions that have been made, and I can show those.
Thank you. Cliff, go ahead.
Okay, last one. So Anthony Purcell again for $5 Australian.
No, not the last one.
Last one on
Yeah,
here.
no.
Harmful/useless viral insertions are not selected for. They are selected against. So the fact the insertions we see are useful is exactly as predicted.
Nothing.
Was that a super? Yeah, I would say a lot of that was addressed in the discussion. I don't want to be redundant. I would just say go rewatch the discussion, which I thoroughly enjoyed, mind you.
It's great. Just quickly, the last few from Snark to be read at the end. Thank you. From Robert, $4.99 for the AC fund. Thank you, love. Good. Let's see. I don't know if you can see any of these, Cliff. That's why.
But you have snow now, Marvel. Why do you need an AC?
It's in between. I live in Colorado, so we have hot days and then snow, and then hot days and then snow. And it's Colorado. Wait 5 minutes if you don't like the weather. It'll change. Craft Tequila for €2. Fantastic debate all around. I concur. Thank you guys again.
I appreciate it. I agree. I concur as well. It reminds me of that. What was that movie with Leonardo DiCaprio? Where he pretended to be a. No, that's a good one too. I think because I'm getting so burnt out for some reason I can't. It's with Tom Hanks.
Catch Me If You Can.
Catch Me If You Can. Yeah.
Catch Me If You Can. Oh, that's a wonderful movie. Yeah.
He watches like some videos on it. I concur, but then he passes out after.
Yeah.
Okay, John Rapp for, I believe that's Australian, $5. Oh, an AC fund. That sounds like a worthy thing. Good day, Marvel Girl. You need to chill? That's ironic. You're so cool. Aw. Good day, everyone, too. He said, hi, everybody. Let's see. Snake was right again. Standing for truth, Snake is saying, for $5. You didn't answer how epistasis can decrease fitness and still not be visible. Also, humans and chimps have a lower CO1 difference than cats.
Right. Yeah. If we would have got into that too, like, for example, God created two humans, Adam and Eve, so we predict low genetic diversity. Lo and behold, that's what we see. That's why the evolutionists invented the out of Africa scenario to explain the low genetic homogeneity. God, according to the Bible, created populations of animals. So we'd expect a higher genetic diversity, of course. This is kind of just common sense. And synergistic epistasis, when it comes to the fitness, he needs to define fitness, because often fitness is defined in a narrow sense. The best definition of fitness should be total functionality. Yeah, Lenski experiment, for example, there were some increases in fitness in a narrow sense, but overall they're getting rid of genes short-term and it's long-term degeneration. So the total functionality has decreased. It all comes down to net gain versus net loss, and the evolutionists have failed in explaining the net gain versus the net loss, because a lot of these genomes, they are shrinking. The Lenski bacteria, they now have shrinking functional genome sizes. This is degeneration at work. So good question.
Okay, great. Stringer News, $10, even though there's snow outside. AC fun. See, he agrees, Raj. And that's it. Thank you. Wow, we made it.
We made it. Good question.
If I missed any, I'm sorry. But wow, you guys really, you were so polite, but so much information. See, that's what happens when you're not like stopping to call each other names. So much more information comes through, you know?
Oh yeah, calling each other names and getting like that is not profitable by any sense.
So anything less to say, either one of you, please, if you want to say anything and sign off, and then I'll end the stream because I don't want to keep you anymore.
I'll just say I'm definitely up for doing this again, but given that, like I said before, that we probably agree, you know, that evolution. Let's not talk about genetics next time. Let's talk about maybe radioactive dating, the date of the Earth, something like that.
Yeah.
Different.
I would say we could title it, because then we can talk about radioactive decay, and then I'd go into, of course, Noah's flood and catastrophic plate tectonics. So this is another topic I'm fascinated with. But if we titled it like, let's see, something like, does the evidence support a young Earth or an old Earth? Because yeah, we can talk about radiometric dating, but I can also utilize because normally I looked at genetics to prove that we at least have a young genome. Right? You could arguably say that Adam and Eve came about 6,000 years ago, and then the Earth itself was billions of years ago. But I think that would be a good title for the debate. Then we can still talk about a number of evidence. Yeah, that's a good idea.
All right, sounds good.
I will be in touch with both of you then.
Brilliant.
Thank you very much.
Good. Catz, I really appreciate it.
It's been good fun. I've enjoyed it. Thanks, mate.
No problem.
And
I'll probably premiere this on my channel in the next
awesome
couple hours for anybody watching. And then we have a debate on my channel tonight. I'm not sure if his name's Mark Drysdale. I've debated him. Kent's debated him a few times. He's debating on intelligent design, John Maddox. So like you put on debates. I like to try and put on good debates as well.
Okay, great.
Good channel. Keep it up, a Marvel girl.
You too.
Thanks for being so civil, guys. I didn't even have to step in.
Oh,
once.
yeah.
Yeah.
I know, right? It was great. And Raj, you've done like amazing job, hasn't he? He's been great. Thank you. Keeping up with everything. I would never have gotten through this without you.
It's still a bit messy though. I saw someone saying that the cartoon is awesome, but it needs to be a tiny bit smaller on the bigger screen, so the bigger screen is a bit more visible. But I did zoom in, so I compensated for that.
Yes.
It's not the size that matters. Oh wait, that's something else.
Oh,
I do have
so used to saying that. No, okay.
She said it again. She started with you, Catz, and now she starts with me. But I do have a YouTube player on the table. If you look, I can't send the audio to you guys because I am missing one channel because I'm using Firefox right now, and I can't send it to Firefox itself. So I can play that if you want, and then listen to it on the stream itself. It's the.
Awesome.
You weren't prepared for that.
Weren't prepared for that. Okay, yeah, that was great. Catz always is so.
The song is playing, by the way. Yeah.
Okay, cool. All right, well, just want to say thanks again. Shout out to the Patreons for helping me put this all together. With your support, I couldn't do any of this.
Radiometric dating.
And thanks for coming.
For that.
Anybody else want to say goodbye?
Bye-bye. See you later.
For all those who appreciate the work that we're doing here on Standing for Truth, please hit that subscribe button because we are just getting started.